Okay. So thank you, Sonia. So the next speaker will be Andrea Haase from Germany, and she will switch to a different organ and different particles again. So it's the brain, the central nervous system, and silver nanoparticles.
Okay, so thanks to the organizer that I've been given the chance to present the results of another study that has been recently performed in our institute. So we were interested in the effects of silver nanoparticles in neural cell cultures. And for that purpose, we applied a nanoparticle system, silver nanoparticles that have been stabilized by a covalent peptide coating based on 3 amino acids, cysteine, lysine, lysine. It's a covalent linkage between the sulfur group of the cysteine to the surface of the silver nanoparticles, and these nanoparticles offer several advantages. So the size and the morphology of these nanoparticles can be very tightly controlled, as you can see in the TEM picture. I cannot pay attention to every small detail of the study, so I would like to refer you to the publications that have been made, so especially concerning the synthesis and characterization. We applied different techniques like TEM, SEM, AFM. For the solution, we use DLS, we use SAX, we use nanosite, and so on. But you see here in the TEM picture that these nanoparticles indeed are very homogeneous. The polydispersity index is very low compared to conventional citrate-coated nanoparticles, as you can see here. For comparison reason, we included a larger size of these silver nanoparticles, 40 diameters in size, with the same peptide coating, and we also included gold nanoparticles with the same size and the same coating. Our cell system that we studied was a mixed primary neural cell system we obtained from cortexes of newborn rats. So 1 to 3 days after the birth, we removed the cortexes and we did a primary cell culture. These cell cultures consist of astrocytes and neurons mainly, but they also contained a few oligodendrocytes and eventually a few microglia cells. So these cells were kept in culture for different periods of time, for 1 week, for 2 weeks, for 3 weeks. And what is going to happen is that the cells grow extensions, the synaptic contacts form, and so after 1 week, basically, you see extensions from these cells, and the synapses just begin to connect the cell-cell contacts. And after 2 weeks, they are fully functional. And so we studied cytotoxicity of the different silver nanoparticles. As you can see here, there is a quite dose-dependent response for the 20 nanometer particles and for the 40 nanometer particles, and the doses are given here on basis of micrograms per milliliter, but we also calculated 2 other dose metrics, and you see here that based on the same mass dose, the bigger particles were less cytotoxic, and the gold particles hardly evolved any cytotoxicity at all. And I would like to draw your attention in the lower dose range that you can see here. This is the different times in culture. So especially in the low dose range, we see that with increasing time in culture and with increasing differentiation, these cultures get more sensitive towards the treatment with the silver nanoparticles. This is not the case for the higher dose, it's just cytotoxic regardless of the age of the culture. What we did then was we analyzed the oxidative stress behaviors. So here you can see the induction of Hem oxygenase 1. It's a well-established stress marker under the control of the NF2 pathway, and you see with the silver particles after different times in culture, after 8, 12 and 24 hours, HO1 enzyme is induced, but this is not the case with the gold treatment, and we see a formation of protein carbonyls, and this is an in vivo imaging of the ROS formation directly in the living cells. We use the dihydroethyllium dye, and we measured in each preparation approximately 30 cells, and we repeated this several times, and you see here, if you treat the cells with silver, you see an increase in ROS production, and this is just a quantification. If you compare now here the smaller nanoparticles, you see there is a dose-dependent increase in ROS production, The bigger nanoparticles also induce ROS, but to a lesser extent. And here is ionic silver, and here we even overestimated the amount of ionic silver that could be released from these nanoparticles. And you see it is insignificant ROS production there compared to control cells, but it's not as much as you see with the nanoparticles. Furthermore, we analyzed calcium responses in this mixed cell culture system. So this is an acute response, and astonishingly, even a few minutes after addition of these particles, so depending on experiments, so 3-4 minutes after addition of the particles, you see calcium response if you treat the cells with silver, but not if you treat the cells with gold. This is just a quantification. I will not go in every detail. It's the area under the curve of all these cells. It's a live cell imaging experiment again, and in each assay you analyze approximately 30 to 50 cells. You repeat this experiment several times, and we also repeated the experiment not only in the mixed cell culture system, but in the pure astrocyte culture and in the pure neuronal culture, or neuronal enriched culture that you get after treatment of the cells with arabinosid C. What you see here is that if you pre-treat the cells with antioxidant, and in the picture here you see N-acetylcysteine, but we also used other antioxidant, you can completely block the ROS production, but you will have no influence on the calcium response. We furthermore studied the uptake of the nanoparticles by TEM, and we did this in the single culture system, so in the pure astrocyte and in the pure neuronal system, but also in the mixed system. And what we see is basically in the astrocytes, we see the nanoparticles, the silver nanoparticles. So due to the heavy load of nanoparticles in these vesicular structures, there is a disturbance in the film during exposure. So we see the nanoparticles in vesicular structures, and we see several of them in one. But for the neurons, we hardly see any nanoparticles in the cells. And we see them in places like here, where it's difficult to say whether that's truly inside the neurons or not. We are not completely sure. But only very few nanoparticles can be seen in that case. Last but not least, we did some kind of immune fluorescence studies, and here you see the morphology of the cells. It's a mixed cell culture system again, and you see the neurons stained in green, you see the astrocytes stained in red, and you see nicely how these cells form networks. Yeah, this is a control cell without any treatment, and if you treat them with nanoparticles, of course they look different, and both cell types are affected. But what you can easily see that the astrocytes are much more affected than the neurons. And also we can confirm this by cytotoxicity studies that I have no time to show today. When you do a cytotoxicity study with a pure astrocyte culture, the response starts much, much more early. With a much lower dose, you see a cytotoxicity for the astrocytes, but not for the neurons. So to sum this up, we see cytotoxicity, which is dose-dependent and dependent on the particles that we apply, on the particle size. And especially in the low dose range, we see a dependence on the differentiation state of the cell. The further differentiated neuronal culture were more sensitive. We have analyzed oxidative stress via different techniques. We can image directly the ROS production in these cells. The ROS production can be blocked by pre-treatment of those cells with antioxidant, with different antioxidants. We have seen acute calcium responses a few minutes after addition of silver, but not after gold nanoparticles. And here antioxidant pre-treatment had no effect. So surely this calcium response is not a consequence of the oxidative stress formation, and also to detect the oxidative stress formation we have to pre-treat the cells with the nanoparticles for a longer time period. You see no acute ROS production. If you treat the cells immediately and measure, you see nothing. If you pre-treat the cells only for 10-15 minutes, you see no ROS. You really have to pre-treat for a minimum of 1 hour, and then you can follow the ROS production. And we study the uptake, and we can show that mainly the astrocytes take up the nanoparticles. Neurons rarely do so, if at all. We are not sure about this. And if you look in the morphology, we see the astrocytes are much more sensitive towards the silver nanoparticle treatment. And this is especially interesting since in brain situation you have many disease situations where it's mainly an impairment of the astrocyte. And because of the loss of the astrocyte function, you get brain impairment. In many neuropathological diseases, you have this disease development mechanism. And so finally, I would like to thank my co-workers. These are the material scientists who produced the nanoparticles. We got support from a TEM team at Free University of Berlin. Also, these are material scientists, and we have support from Eurobiochemistry department in Magdeburg. And finally, I would like to draw your attention to the references. So thank you very much.
Thank you very much, Andrea, for the presentation. Some questions from the audience?
There is a question on my side. If I may, do you see a difference in sensitivity to the silver nanoparticles between dopaminergic neurons and catecholaminergic neurons? Does the presence of neuromelanin, that is a scavenger of toxic substances, make a difference?
The difference, sorry, can you?
Between dopaminergic neurons and
yeah, that's a very interesting question. We will continue the studies. We didn't check for this in this phase of the study, but we will extend the study, and that's a very interesting point to follow. Yeah. No, we have no answer yet.
I may have a question if nobody else.
The free calcium that you were measuring in the cells. It takes quite a while for that to get going in the cells compared to, say, a rapid calcium spike. Do you think that's just membrane permeability changing, or ROS releasing intracellular stores? What do you think's happening?
I expected this question somehow. We cannot answer. We will look for the mechanism. My personal guess is it's from the extracellular space, and since we also see this with this speed, I mean a few minutes after addition, and we know from silver that silver has a high affinity for sulfur-containing protein, I would assume that maybe it attaches to the outer membrane of the cell to some channels and does trigger the calcium response. But we have to analyze. I cannot say.
Michael?
Very interesting study. I wonder about how much you know about the oxygen pressure, partial pressure in the brain compared to the oxygen pressure you used in your studies. Did you control anything to be in the same level or the comparable?
We are in the same level as you would expect for a healthy situation, but the group we are cooperating on, which is the Neurobiochemistry department in Magdeburg, actually they are not nano specialists. So I brought in the nano topic to their research. They are specialists for ischemia. And so they are specialists for this specific question, hypoxia and so on. So we didn't explicitly connect to this nano study, but we might do in future.
So I think we have to stay on time, more or less. So thank you very much, Andrea.